Hello, welcome to my channel. So today we are continuing with the human anatomy and physiology core lectures. So today we are going to be talking about transport across the cell membrane. So we are still on cells, so we are going to spend some time on cells. Okay, so as we learn the anatomy and physiology of cells. Okay, so in terms of movement of molecules of cells, there is a passive transport, we have active transport. We have endocytosis, where we have phagocytosis and pinocytosis. We have exocytosis. Okay. Then the transport across the plasma membrane, going now in details. So we have passive transport, which does not require energy, and they move due to a gradient. It is a difference in concentration, pressure, and charge. Then they move to equalize gradient. I moves towards low. Okay, then types of passive transport, we have diffusion, osmosis, facilitated diffusion. Okay, then going into details on simple diffusion. This is when molecules move to equalize concentration without the help of membrane transport proteins. Okay, and this is an example of a simple diffusion where it's the movement of particles from high to low concentration. Okay, so as you can see, this maintained this diffused evenly whilst maintaining an equilibrium. Okay, and we use the dye molecule and water molecules. Then for osmosis, this is in osmosis, the water moves through a selective permeable membrane from an area of lower solute concentration to an area of higher solute concentration through a selective permeable membrane. Often involves movement of water into cell and also of course out of cell. Okay, so this is an illustration of osmosis. As you can see, during the initial state, the solute particles were partly shield membranes pause for the passage over solvent molecules from right to left. So the solvent molecules were here, then the solute molecules were here. Then after net movement of solids through a semi-permeable membrane, the osmotic pressure was like this, and this was the final state, and there was movement of molecules from I from low to I. Okay, that was the movement from higher solid concentration from low solid concentration to an area of higher solid concentration. So meaning, if you look at the solvent molecules in this area, it was low, and in this area it was high. So the molecules moved from low and they went into the high section through the selectively permeable membrane. That's osmosis. So from low to high. Okay, then in terms of solution of differences, so a solvent plus a solute, that's a solution. Then we have an hypotonic solution where the solids in cell are more than that of the outside. Then in hypotonic solution, outside solvent will flow into the cell. Then in isotonic solution, the solids are equal inside and out of cell. Then in hypertonic solution, the solutes are greater outside of the cell. So the fluid will flow out of cells. So since you are going from low to high. Okay, and this is an illustration of hypotonic and isotonic and hypertonic. So in a hypotonic solution, the dilute we move from low to high. So the high in this case is the cell, and the cell of illustration we are using a red blood cell. Then a isotonic solution where everything is equal, there won't be any net movement. Then in an hypertonic solution, where the solution is where the cell is one which is having low solutes, so the solutes are going to move from the cell to the outside, and the cell will become crenulated or shrunken. Okay, then let's go to facilitated diffusion. So differentially, permeable membrane and channels. We use channels that are specific to help molecules or ions enter or leave the cells. So channels are usually transport proteins, so meaning aquaporins that facilitate the movement of water. Then no energy is required or used. Then in facilitated diffusion, a substance moves across the lipid bilayer aided by a channel protein or a carrier protein. Then processes of facilitated diffusion, so the proteins bind with molecule and the shape of the protein is going to change and the molecule moves across the cell membrane. That's what the facilitated diffusion is. Then in terms of active transport, this is more of a molecular movement. So a molecular movement requires energy against a gradient, and example is the sodium potassium pump. And the types of active transport include the primary active transport and the secondary active transport. And this is the illustration where we have low concentration of molecules or ions outside the cells, which moves against a gradient using energy. Okay. Then in terms of going further into primary active transport, so in primary active transport, the energy derived from the hydrolysis of ATP changes the shape of a carrier protein, which pumps a substance across a plasma membrane against its concentration gradient. So indeed, the carrier proteins that mediate primary active transport are often called the proteins, the pumps. Then the most prevalent primary active transport mechanism expels sodium ions from cells and brings potassium ions in. So because of the specific ions it moves, this carrier protein is often called the sodium potassium pump. Okay, then the secondary active transport So in secondary active transport, the energy stored in a sodium ion or hydrogen ion concentration gradient is used to drive other substances across the membrane against their own concentration gradient. So because a sodium or hydrogen ion gradient is established by primary active transport, secondary active transport indirectly uses energy obtained from the hydrolysis of ATP. Then we have two things which you need to know, that we have antiporters, which carry two substances across the membrane in opposite directions. Then we have symporters that carry two substances across the membrane in the same direction. Okay, so this is more of the differences between the passive transport and the active transport. Okay, you can pause and even go through. Then we have other types of movements, which is the endocytosis. So this is a movement of large material, such as particles, organisms, large molecules. Movement is into cells. Then you have three types of endocytosis. So we have receptor-mediated endocytosis, phagocytosis, and bulk phase endocytosis. Okay, so these are the types of endocytosis that we have. So the phagocytosis, pinocytosis, and receptor-mediated endocytosis. And the bulk phase endocytosis, otherwise known as the fluid endocytosis, and bulk phase pinocytosis. So pinocytosis is just another name for the bulk phase endocytosis. Then processes of endocytosis include the plasma. You have to know that the plasma membrane is going to surround the material, and the edges of the membrane meet, and the membranes are going to fuse to form a vesicle. So that's why some people even call this vesicular or vascotransport or vesicular transport. Okay. Then the other type of transport is the exocytosis, which where there's a reverse of endocytosis, and which is more of a reverse of endocytosis, and the cell discharges material, and the vesicles move to cell surface. Then the membrane of vesicle fuses, and the materials are expelled. So this is a reverse of endocytosis, and this is the illustration. Okay. Then the last type of movement is transcytosis. We are in this active process called transcytosis. Vesicles undergo endocytosis on one side of a cell and move across the cell, and then undergo exocytosis on the opposite side. So as the vesicles fuse with the plasma membrane, the vesicular contents are released into the extracellular fluid. So transcytosis occurs most often across endothelial cells that line blood vessels, and is a means for materials to move between plasma membrane and interstitial fluid. So for instance, when a woman is pregnant, some of our antibodies cross the placenta into the fetal secretion via transcytosis. That's the mode of transportation. Okay, so that's the end of the types of movement, and together the end of the tutorial itself. And I thank you for watching. If you haven't subscribed, please subscribe and hit the bell notification. And of course, see you on the next anatomy and physiology lecture tutorial videos. And if you haven't subscribed, please subscribe, and also check out the anatomy and physiology playlists as well. Okay, so give us a like as well.